Have you ever stopped to wonder what's hiding in plain sight inside a single drop of pond water? It is a pretty simple question, but asking it completely changed the course of science. It opened our eyes to a literal invisible universe, a world of microscopic organisms that live, feed, and multiply all around us, and, even inside us.
Welcome back to The Explainer. Today, we're diving straight into the microcosmos to demystify microbiology. We're going to trace its roots all the way from the very first primitive lenses that revealed these tiny life forms, right up to the grand biological filing systems we use today to organize all this unseen life.
Here is our roadmap for today. We'll cover the invisible world revealed, pioneers of the microcosmos, meet the microbes, classifying the unseen life, Kingdom Monera, the prokaryotes, and finally, Kingdom Protista, the eukaryotes.
Okay, let's jump right into Section 1, the invisible world revealed.
Think about it. For almost all of human history, if you couldn't see something with your own two eyes, it simply didn't exist, right? But that all changed with the invention of the primitive compound microscope. Before this ingenious tool, microbiology was literally impossible. But by cleverly combining lenses, early scientists were finally able to see things so much smaller than the naked eye could ever perceive. It was a total game changer.
Moving on to Section 2, pioneers of the microcosmos.
So how did our understanding actually build over time? Well, back in 1665, the English scientist Robert Hooke made the first observation of cell walls. Then, just a few years later in the 1670s, Anton van Leeuwenhoek actually observed living cells swimming around in pond water. Fast forward to 1833, Robert Brown discovers the nucleus in plant cells. And all of this sequential discovery culminated in 1839 when scientists jointly proposed the unifying cell theory. And that's what laid the groundwork for later pioneers, like Louis Pasteur, to definitively prove that microbes are the engines behind things like fermentation and disease. Consider Robert Hooke's famous 1665 sketch of a slice of cork. Since cork is just dead wood, Hooke was technically only looking at dead tissue. But the honeycomb pattern he saw, these tiny, empty little rooms, is exactly what prompted him to coin the term cell for the very first time. He was literally just looking at the rigid cell walls left behind.
Which brings us to Section 3, meet the microbes.
Now, while Hooke was staring at dead walls, Anton van Leeuwenhoek was discovering living miniature animals, like the amoeba. This is a tiny single-celled organism you find in the mud of shallow ponds. It's incredibly small, about 1/4 of a millimeter, with this bizarre, irregular shape. And what's really fascinating is how it moves. It sludges through the mud using something called a pseudopodia, which literally translates to false foot. It just extends this gel-like foot to pull itself along the bottom. And then you have Volvox. This proves that single cells don't always fly solo. They can actually team up. Volvox is a type of green algae that forms these massive, beautiful spherical colonies of up to 50,000 cells in freshwater habitats. And yes, Leeuwenhoek was the very first person to report seeing these, way back in 1700.
Let's look at Section 4, classifying the unseen life.
Okay, so you have all these crazy microbes being discovered, but science quickly ran into a massive logistical problem, naming them. Common names vary wildly depending on your language or region. Like the common onion is called pyaz in Urdu, but ganda or basil elsewhere. Imagine the utter chaos of trying to identify thousands of microscopic organisms that way. That's why the Swedish naturalist Carolus Linnaeus stepped in to establish a universal scientific naming convention, making sure one accepted name applied to only one species, universally.
Fast forward to the 1930s, and the electron microscope comes along and shakes things up again. It revealed two incredibly distinct microscopic patterns, kicking off a major biological sorting process. Scientists realized some organisms were super simple. They called these lower protists, which were prokaryotic, like bacteria. Others were much more complex, termed higher protists, which were eukaryotic, like fungi and certain algae.
That perfectly sets up Section 5, Kingdom Monera, the prokaryotes.
That clear structural split basically forced scientists to create a brand new filing cabinet just for the simplest life forms, and that became Kingdom Monera. Think about Salmonella, for example, with its bright, pill-shaped structure. It's a perfect representative of this kingdom. They are rugged, they're incredibly small, and structurally, they're actually quite simple on the inside compared to, say, human cells.
So to summarize Kingdom Monera, it is strictly VIP access for the prokaryotes.
These are the unicellular organisms, the lower protists, that entirely lack a complex, organized nucleus. This kingdom is the official scientific home for all bacteria as well as cyanobacteria. If it's a simple, single-celled organism without a true nucleus, it belongs in Monera.
And for the rest, Section 6, Kingdom Protista, the eukaryotes.
What about the single-celled organisms that actually do have complex internal structures and a proper nucleus? Well, those get filed under Kingdom Protista. Think of this as the club for eukaryotic single cells, organisms that kind of straddle the line, having traits of both plants and animals. They're mostly aquatic, including protozoa like our old friend the amoeba and the paramecium, plus unicellular algae. But here's a fun caveat: it specifically excludes yeast, even though yeast is single-celled.
Modern biology relies heavily on the five-kingdom system to draw strict boundaries, and it really comes down to just two major things: cell type and mode of nutrition. Monera is prokaryotic and feeds via photosynthesis or absorption. Protista steps it up to eukaryotic and adds ingestion to the menu. And then you have fungi, which is eukaryotic but strictly feeds by absorption. And speaking of fungi, remember how yeast got kicked out of Protista? That's because fungi snag their very own kingdom. They're totally unique because they have cell walls made of chitin, and they are what we call achlorophyllous. That's a big word, but it just means they completely lack chlorophyll. Since they can't make their own food through photosynthesis, they are absorptive heterotrophs. They literally have to secrete enzymes and absorb their nutrients directly from their environment.
It really is amazing to think about. From Hooke's primitive lenses looking at dead cork walls to modern electron microscopes mapping the intricate differences between Monera and Protista, our understanding has just exploded. But as we wrap up this explainer, I want to leave you with this. Knowing that science is still mapping this massive microscopic universe today, what bizarre, totally unclassified eukaryotic and prokaryotic colonies are thriving entirely unseen right on your hands at this very moment? Think about that for a second. Keep learning, and I'll see you in the next explainer.